Hello and welcome everyone to this podcast. In this podcast we're going to focus on the introduction to inheritance. And let's begin with what we talked about at the very beginning of the semester when we gave a brief definition of genetics. And we said that genetics is the study of heredity and study of the variation of traits. We've talked a little bit about heredity, but up to this point we've talked mainly about the variation of traits. So remember, we began this semester talking about simple nucleotides and how these nucleotides were constructed in such a way to make DNA. And we talked about the properties of DNA and how it formed and the history of discovering how it was the genetic material and all this great stuff about DNA. We also talked about how it was replicated through replication. And then we also talked about how through the process of transcription we would make RNA. And then how this RNA through the process of translation, TLN, we would make a protein. And how this protein really was what we call the trait. Yeah, we did talk a little bit about how RNA sometimes is the final product, and that is the trait that we're interested in. But the key is that DNA somehow or another makes a trait. More recently, we talked about how DNA could be mutated, and how when it was mutated, we would get an altered RNA, which I'll put a star by because it's altered, and then how that would lead to an altered protein. Or I should say, could lead to an altered protein. Sometimes this would be a new trait. Sometimes it could be a non-functional one, or we'll put disease trait. And we talked about how these changes in the DNA that made these new traits could lead to evolution. Or it could help explain how evolution has occurred. And so this is due to mutations. I should put it all the way with the DNA. Because remember, the mutations occur in the DNA, but they affect the RNA and protein. Now for this next section, what we're going to do is we're going to talk about heredity. How the DNA is inherited. I'm going to write heredity. Inheritance, heredity, pretty much the same thing. And how through the process of heredity, we pass on this DNA to offspring or progeny. And how these progeny, having this DNA from their parents, will have traits similar to their parents. But we'll see not exactly like their parents, but similar. And how when these mutations occur, how that affects heredity and ultimately the progeny. We're going to talk about some old ideas of heredity. We're going to talk about our more modern view of heredity. And we may even talk a little bit about how some of our modern views of heredity have changed our minds about the old way of thinking about heredity. Biology is a fairly young science, and genetics is even more recent. And because of that, it makes studying genetics a little confounding, because we often have to change the rules, because we're learning more and more about genetics all the time. So this will take us through the next several days. And just to kind of keep your eye on the future, we're going to close the semester with thinking about how all of this, how we make traits, how genes are passed on to our progeny, how mutations affect this process, and we're going to close with our understanding of how all of this affects populations and evolution. But first, we're going to focus on heredity. And let's talk a little bit about history, the history of heredity. When we start thinking about the history of heredity and the history of genetics, we could go back, if we would like, all the way to 10 to 12,000 years ago. And this is because all these thousands of years ago, plants and animals started to become domesticated. And once that happened, the people of the time were interested in how to preserve certain traits or how to select certain breeding types in order to give plant progeny or animal progeny that was needed. So I'm going to write domestication. Now, they didn't know what they were looking at. They didn't know there was DNA in there. They didn't know a lot about this. In fact, they knew very little about the finer details of the cell and of the genes. That would come thousands of years later. But they kept thinking about it. And there's a lot of time I'm skipping here. But let's go back now to about 400 to 300 BCE. And two people, Hippocrates and Aristotle, came up with this idea. And the idea, we'll call it Pangenesis. So let's go ahead and write that here. The word Pangenesis was a term that ultimately Darwin came up with, but it was based on the ideas of Aristotle. And the idea of Pangenesis, let's see if I can make some attempt of a drawing of a human here. So the basis of Pangenesis says that every cell in our body, every tissue in our body, produces these very tiny particles. And these tiny particles, so let's just make some particles here, in all of the tissues of our body, they all send these particles from the different parts of our body to the gametes. And that's how, when we have progeny, or they reasoned that when we had progeny, that they ended up looking like the parent. So similar progeny would result because of this. Now, this idea of Pangenesis brought about this other idea, and it's linked to Pangenesis. And it's something we're going to call the inheritance of acquired traits. And all this says is that anything that this adult here acquires throughout their life, if they learn how to play the piano, if they learn to be a great baseball player, if they learn the best place to go fishing or hunting, all of those traits are going to be passed to their gametes and passed on to the child. Inheritance of acquired traits. So anything they acquire throughout their life and before they have their child is going to be passed on to them. So this is not exactly what we think today, but they were slowly building upon what they had observed. So as we move forward, I want you to think about this question here. What observation have you had that refutes this idea of pangenesis or inheritance of acquired traits? What have you experienced in your life that tells you that the things that this adult acquires throughout their life isn't necessarily passed on to their child? Okay, so in the early 1600s, there was this idea called preformation. And in this idea, there was this little structure called a homunculus. So this homunculus, which I should say stands for as a little man, said that there was this tiny individual, tiny human, so there's a tiny human inside the sperm. That was the prevailing thought. And so these individuals were called, cleverly, spermists. There were some also, not as many, but some that thought that the tiny human lived in the egg, or the oocyte, and they were called ovists. Almost sounds like parts of an orchestra. But the ovists believed that the tiny human was in the egg, and the spermists believed the tiny human was in the sperm. And here's a picture of the way that they thought the tiny human and sperm would look like. And they believed that everything was fully formed in this tiny little human in the head of the sperm. And all it had to do was get inside of the egg. And once inside of the egg, it would give it an opportunity to grow. And the ovists believed something similar, that the tiny human was in the egg, and somehow or another the sperm, when it fertilized the egg, that it would cause it to start to divide inside of that egg. Or I should say divide, I should say mature and grow. One of the major flaws to this idea was that if this were true, if the tiny human was in the sperm or in the egg, who would the progeny look like? And so in this case, if this tiny human was made in the male, inside the sperm, the prediction would be that all the progeny are going to look like the males, and none of the females will have any of those characteristics, and vice versa for the ovists. Well, we know this isn't true. We can look at our own families and see that, yeah, some of our traits look like mom, some of them look like dad, some of them look like a great-great-grandfather, great-great-grandmother, right? There's no necessary male or female side that we resemble more closely. Now I want to jump into the late 1800s to early 1900s, 1890s I should say, to 1910. This general time frame. This happens to be after Mendel did his work, but we'll talk in a moment how much of Mendel's work was left undiscovered until the early 1900s. But it's important to note what this German scientist by the name of Weizmann, August Weizmann, came up with that refuted Pangenesis, the inheritance of acquired traits, and this idea of a preformation event. And he came up with this theory called the germ plasm theory. And in this theory, it states that all of the characteristics that are to go on to the next germination reside in the gametes, both gametes. So both gametes have the characteristics, abbreviated here, to form progeny. And it didn't really matter what that organism learned throughout life or the skills they acquired, because all of that was in their gametes, and it was not influenced by somatic cells. That's a really important part that I should put here. So no influence by the somatic cells. So Weizmann did this interesting experiment where he took a mouse that had a tail here, and he chopped off the tail, and he let this mouse make progeny, and this mouse had a tail. And he did this experiment over and over again. So this mouse cut off the tail. The mouse that was born, the progeny, had a tail. And he did this about for 20 generations. And that 20th generation, the mouse that came out, shockingly, had a tail. And the reason for this is because the idea of acquired traits, inheritance of acquired traits, isn't necessarily true. That what you do to this mouse doesn't necessarily affect the progeny. So at this time, it was pretty well accepted that the germ plasm theory is true. That everything that would help to determine what the next progeny would look like, those genes, those traits were all determined by what was already in the gametes. And it didn't matter what happened to this mouse throughout its life. And we've already talked about this, but in case it's like bugging you, it's popping in your mind, and you're wondering why I'm not addressing it, is our good friend epigenetics. If you remember, I said that at the beginning of this podcast that genetics is a very confounding subject to study because we keep changing things. Every now and then new evidence comes along and says that, well, maybe the way we thought about things that initially aren't true anymore, or aren't the best explanations anymore. And though we certainly support the germ plasm theory, and certainly what you do to this mouse by cutting off its tail or removing its fur or some kind of physiological or anatomical change isn't going to change the way this animal is going to look. You can look at it the same in humans. If a child loses their arm as a child, or an adult loses an arm as an adult, and they have children, their children will still have arms. That trait is not going to be passed on. We do know that there are some things now, because science continues to expand and grow and build upon knowledge, we do know that some of the things that an organism experiences in their life can be passed on. So there are certain exceptions to this rule. And what kind of rule would this be if there weren't exceptions? So there are exceptions. But for our purposes, if you're asked on a test, what is the current theory that is supported for inheritance, you would certainly say germ plasm theory. Okay, I'd like to jump back a few years. Before Weizmann did his experiments, there was this monk in Austria, Gregor Mendel. And here's a picture of him. And down here is the garden that he worked in as a monk. This is where he grew his peas. But this is what Gregor Mendel looked like. I often think he looks like Kelsey Grammer from Frasier. But it is Mendel. So a few things about our friend here, Mendel. He grew up in a poor farming family in the Czech Republic. And one of the ways to have your child become educated in those days was to have them enter the seminary. And so that's what happened. He entered the seminary. He's a pretty smart kid, and he would then become a priest. He always had a passion for both two things: botany and math. He didn't know he was a geneticist or that he would become a geneticist. In fact, on his deathbed, he probably didn't realize he was the father of genetics. In fact, he certainly didn't know he would become that, because much of his work was largely unrecognized in his life. He made his studies, his revolutionary studies in genetics, in Brno, B-R-N-O. And that's where he did his pea studies. He published his works in an obscure journal in 1866, but not many people read this journal, so it did not really spread very quickly. Interestingly, around the same time, Darwin was making his discoveries aboard the Beagle, the ship, the Beagle, as he was traveling around the world picking up samples. If the two of these people would have ever met, Mendel and Darwin, they probably would have had some great discoveries together, because they were both on the verge of bringing these two ideas of genetics and evolution together. Shortly after these studies, he became the abbot, which, from my understanding, is the person who's in charge at the monastery. So he took on a larger administrative role, and he died in 1884. And his work remained undiscovered, largely undiscovered, until 1900, when three scientists, Vries, Schatzmark, and Correns, and we may talk about the three guys a little bit later, but they went on to rediscover his work, and to their credit, at least two of their credits, they recognized Mendel for his original discovery of this work, as opposed to just taking credit for themselves. Alright, well, that's a little history of Gregor Mendel, and now let's talk about his peas. Why did it turn out that the peas were an ideal organism for him to work with? These are many of the same ideas why any model organism is advantageous. These peas, any given pea plant, produced many progeny, up arrow for many progeny. They were relatively easy to grow. You need some seeds, you need some soil, and you need some water and some sun. They were relatively inexpensive. You could go down to the market and buy pea seeds. So it wasn't very expensive. We'll say a reasonable generation time. They're plants, so he had about a 1 year generation time. Now, for modern science, this is really not a very reasonable breeding time, but for him it worked out well. He had lots of time, so it didn't really affect him too much. One unique thing about plants, unlike some model organisms, you can cross them easily, and you can cross fertilize them. So you can take a plant that is, say, big A from our last slide here, and cross it to itself. So essentially it's a big A times a big A, big A. You can't do that with most animals, right? This is something that is unique to plants. And I'm going to say here, easy to define traits. He looked at things like seed color, flower color, the flower arrangement, the seed shape. Lots of things that were easy to recognize as he looked at the plants. So that made it easy. And the last thing, and I ran out of room here, so let's move Gregor up here. I'm just going to put used math. Many people had made these observations that previously. They knew if you took certain plants, you cross them together, that you'd get some kind of progeny in some kind of reasonably predictable manner. But what he did that was unique is he applied math to it, so he could make predictions about what the progeny would look like. And he was able to establish firm numbers for these, which told us a lot about where these genes were at and a lot about the mechanisms of heredity. Remember, he loved plants and he loved math. This was a perfect situation for him. Okay, let's move on to some important terms that you're going to need for the next several podcasts. So as we move forward with this topic of inheritance, just remind yourself some of the terms, many of which we've already learned, and you just need to make sure you're familiar with them. And I'm going to list them here: gene, allele, locus, heterozygote, homozygote, genotype, phenotype, and pure breeding strain. So remember, a gene is what codes for that ultimate trait. An allele is a different version of the same gene. Locus is the place on the chromosome where that gene is located. So these are all terms we've used in the past. A heterozygote versus a homozygote, we've talked about these as well, but remember, a heterozygote would be something that would have these two alleles, for instance, a big A and a little a. Whereas the homozygote would either be big A as a homozygote dominant, or little a as a homozygous recessive. Genotype, remember, this is what the gene is. So for instance, big A might be the genotype. And if this, for instance, coded for eye color, maybe the phenotype would be brown eyes. The phenotype is the physical appearance that is coded for by the genotype. And pure breeding strain, this is not a term I believe we've used in the past, but this is when you take a strain and you cross it over and over again, and it still appears the same. So after multiple crossings, you get the same genotype. So we could go, and phenotype for that matter. So if you took like this big A, and you had to do, say, 50 crossings over and over again, and you still ended up with big A, then we would say it is a pure breeding strain. You're never seeing anything that looks like little a, which might be blue eye, say, in our example here. Okay, we're going to end this podcast here, and in the next podcast, what we're going to focus on are some of Mendel's early experiments, his monohybrid and dihybrid crosses. All right, if you have any questions, please let me know. See you in class. Bye.